Hey, boys and girls, and whatever.
Talk about barely functional. My brain is going to be a challenge today.
Okay. Yep. Well, that's okay. My brain is going to be a challenge too, actually. No, not so much. Kind of awake. This is my time of the day. Anyway, so the reason for this gathering is I'm having a conversation with a guy named Cebro648 on Rebekah's channel, and also had some conversations with Rebekah about just the story. So linked in the description is a video where she, apparently Rebekah just found out about alternative splicing. And I don't understand that, but she said that in the video that she just discovered alternative splicing, and she should have discovered it a long time ago with the way she presents herself as being, you know, somewhat of a creationist expert in this field. But anyway, aside from that, there's this conversation that I've been having with the man. I don't know where to start. So I know where I should start. Okay. Whoops. What the fuck? What's happening? Do you have any idea what's happening? No, we don't want to do that. God damn it. Hang on a second. Oh, fortunately it wasn't. I still have my camera on. Good. Okay. God damn. I was all prepared. I mean, I'm like really prepared, right? And yet I can't find anything. Son of a bitch. Hold on. Just bear with me here for a minute. Okay. That's not where I wanted to go. Where do I want to go? Yeah, there it is. But it's not working. It's not working. Hold on a second. Let's see. We haven't done this for a while, have we? Can you tell?
We have not. No, we have not. Other than
you can tell. Data. Okay, so the next place I want to go is back here, and I want to show you a gene. Okay, so I'm going to make myself big. Make myself big again. What would that be? MMBA? Okay. Okay. So this is the FXN protein. Okay. It has to do, it goes into the mitochondria and it creates these little clusters that help you metabolize. And if you have a mutation in it, you get a disease called Friedreich's or something like that, and you end up dying young. But anyway, you see these little bars, right? These kind of green bars here and there, right? You see them?
Yes, I see the green bars.
Yeah, or blue bars, right? Those are exons, okay? And these little arrows in between, those are introns. So you can see that exons are really tiny, okay, compared to introns. Introns are really big, and those get spliced out and pretty much thrown away for the most part. According to creationists, they're all, you know, highly functional. But let me see if I can zoom in 10 times and show you a little more of what
You mean creation geneticists that are such a backbone of our genetics
right.
academy?
Yeah, real backbone of okay. So I'm in the introns. I went out 10. Okay, there's an exon. So this part actually gets like translated into protein. Okay, but the rest of that does not. So I'm going to zoom out 10 times again. So I show you the whole gene. So what we have, let me try to explain this. What is it? A get tag? Yeah, get tag. Okay, so basically you've got, why do you do that? I got to set my sensitivity. So you got an exon here, and you got a big intron. Then you might have a little fatter exon, intron. You got another exon, intron, and then maybe a little fatter exon. Okay, so these parts here are sequences of DNA. Okay, right here, and there, and there. And what we want is we want to take all of these and we want to condense them into one big coding data sequence that actually gets translated into a protein. Okay, is that clear? Scott? Scott? Are you with me, Scott?
Let's get right into a protein.
Okay, so this all of this stuff basically gets condensed down here into a coding sequence, coding data sequence, basically. And these parts here, okay, right here, there's a GT usually at the beginning of the intron, and an AG at the end of the intron. And then there's a little, my thumbnail is basically shows you this little tiny sequence right here that becomes the pivot point, right? And so what happens is you've got this exon, you got the intron, and it takes that pivot point and it just splices it out. So it takes that whole thing and splices it and gets rid of it. It tosses it out in the nucleus, and it doesn't just toss it out. It kind of hooks it up into kind of a lariat structure so that it can be recognized and degraded by the machinery in the nucleus. And the nucleotides go back into the nucleus, and they end up being used to. They're basically junk that it throws out, right? So the big fight we're having is how junky are they? Are there any functions in these introns, basically? So am I being clear so far? So these introns they amount to 25% of our DNA, I think, or something like that. Huge amount of our DNA is just introns. Only about 2% is this coding stuff, or 2 to 7% or something like that is the coding part. Okay?
Looks like Austin has deigned to join us.
Hi, Austin. Good to see you. Anyway, so the point is that these things get spliced out with that because they have that little GT AG tag, GT, and then the whole section in the middle. And so there's a particular kind, and it seems very wasteful. But now the thing is that some of these introns are sequences that can cause the gene to be transcribed or repressed. So sometimes these sequences in the middle here have a little bit of function, right? So they do something. And sometimes they turn into like long non-coding RNA, that sort of thing. But now here's the beauty of the introns, okay? If we have copy errors when we copy our DNA, so we copy this whole thing, we want these exons to be preserved because we don't want mutations there. So if you have a long stretch of, say, oh, say you got 30,000 or 100,000 BPs, right? So 100,000 base pairs for one gene, okay, equals one gene. And out of that, 4,000 are protein coding, okay? So that'd be that'd be a protein of about 1,300 amino acids, right? That's probably way off. So let's see. So if we had if we had 1,000 of these out of 100,000, so 1%, okay, we would end up with a 330 amino acid protein. Now, what that does is statistically, if we're going to have a mutation, it's pretty much going to end up in these introns, more than likely. And if we have a sign or a line, like these jumping DNA things that jump in, these parasites, they're more likely going to jump into the middle of an intron and just make the intron bigger. And of course, because of the splicing machinery, it doesn't matter. It gets thrown away and it's junk DNA. So is that clear?
Yes, sir, but don't give me a quiz on it.
I'm giving you a quiz right now. A spot check. Okay. So these introns actually have a function in that they absorb a lot of the mutations that, because they don't matter, because their sequences don't matter. So they manage to statistically absorb a lot of the problems that we would have with mutating DNA. And that allows us to live for about 100 years before things get crazy. So anyway, so that's that. Let's see. Go over here.
Here's a little blurb from Copilot that's helpful. These elements, talking about lines, signs, retroposons, and introners, or something like that, I think is what it's called. These elements are dubbed selfish or parasitic because they prioritize their own replication, even if it harms the host organism or offers no benefit. Does that jibe with what you think?
You're going to hate me. I missed that. Okay, read that again.
Okay. These elements, talking about lines, signs, retrotransposons, and introners, whatever. Introneers? Maybe, I don't know. These elements are dubbed selfish or parasitic. Are you with me?
Yeah.
Because they prioritize their own replication, even if it harms the host organism or offers no benefit. Right.
Okay. Yep. So anyway, the whole discussion is about, and why can't I find my picture? Damn. Where is my picture? Okay, so let's go. Oh, there it is, right there. Okay. So what the hell? Oh, yeah, what Rebekah's whole thing is about here. We got to go back over here again. Okay, what Rebekah's whole thing is sometimes there is a sequence in here that causes this not to splice. So instead of having our protein consisting of that exon and that exon and that exon, it'll skip them and only have 3 exons. So that's called alternative splicing. So sometimes like this one may get cut out and we'll just have 1, 3, and 4 exons, right? And sometimes, we'll just have 1 and 2 and 1 and 4. So we can end up with different proteins from the same gene. And they figure we have about 20,000 genes, and there's estimates that we have about 100 to 200,000 proteins that are created from those genes because of alternative splicing. And that's what Rebekah just discovered. And she says, oh, how can that possibly happen by natural means? That has to be an intelligent designer. And anyway, so that started the whole
And what
caboodle.
is the irrational component of that thought process? Could it possibly be incredulity?
Incredulity is what we got here. So this is the whole thing.
Austin Sims asks a question. It's on the screen.
Well, I don't think she's a flirt or an anti-vaxxer, but she denies all of geology, all of cosmology. What else? She denies. She's a young Earth creationist, so she denies most of science because she knows better. She's a lot smarter than those scientist guys. Those scientist guys, they assume naturalism, which of course Rebekah never does. So, oh, hi, Dave. Dave's here, and Austin's here, and Benghazi's here. We got anybody else that I missed? Did I miss anybody? Okay, so I got into this conversation with this guy. He posted Cebro648. This is his post, or comment. This is crucial in argument against group 2 intron evolving into spliceosome. Okay, so what does that mean? Well, the current idea is that a long time ago, in a sea far away, there were some bacteria about 3.5, 4 billion years ago. And these bacteria ended up getting a virus stuck in the middle of their genome, a transposon, one of those ERVs, those transposable elements, got stuck inside of their genome in the middle of a protein, basically, and it kind of became a problem for these bacteria, this virus that was attacking them, right? So a lot of the bacteria, probably about 99% of them that got the virus died. But 1% of the bacteria had another protein that ended up splicing that thing out. So that pattern, that GTAG pattern that's used for splicing may have been a characteristic of that virus. And so it's called a type 2 intron. And it's really old, and bacteria have them, and are primarily parasites in bacteria. But our hypothesis is that splicing mechanism and those original introns basically became evolved into our introns, and we're filthy with introns, right? There was just one maybe in the bacteria, but we have tons of them. So over almost 3 billion years of evolution in the bacteria, we ended up with eukaryotic cells that ended up with this spliceosome, with this mechanics to get rid of that parasite, right? That cuts them out of the thing so that we can develop a proper protein. And those became our introns, and that became the first eukaryotic cells. Now, this guy says, oh, that can't happen because they're often parasitic. In bacteria, they're parasitic, right? And he's saying that, for our introns are often functional, or they're always functional, according to him. Go ahead.
Quick context check up here. He said that can't happen because it's parasitic. What can't happen?
It couldn't possibly have been the source or the evolutionary stem of our spliceosome. That our spliceosome is very complicated machinery right now. The thing that cuts those introns out.
You mean like 3.5 billion years later, it's more complicated?
Oh yeah, more complicated. And
so before it was simple. After it's complicated, and it's changed over time through populations of other genomes into something else.
What is that called?
There's a word for that. I can't. Maybe Austin or Benghazi can.
Yeah, somebody can come up with it.
I don't know.
Right. Yeah,
it's kind of like the word revolution, except without the R.
Yeah, I don't know. We'll think of it. We're old. It takes us a while to think of these things. Anyway, okay. So he says group 2 introns are tolerated in bacteria, but they're often parasitic rather than beneficial. Many bacterial lineages have lost them entirely because they didn't need them, because they didn't have the transposon in them. They didn't have the parasites, so they didn't need them. They're a far cry from spliceosomes. But here's the thing. Those spliceosomes that are in extant bacteria today have sequence homology with our spliceosomes, with our much more complicated ones, right? So we find the same genetic sequence in these old bacterial mechanisms, spliceosomes, as we do in ours. And that is evidence for evolution. That's evidence that the hypothesis that they evolved from these early type 2 introns, that is evidence for that hypothesis. You see that?
Boom.
Yes. Okay. But he insists that no, because they're parasitic in bacteria, they can't possibly be the same. They can't be evolutionarily related, because in humans, the introns aren't parasites.
Discussion about some sort of teleology in evolution, because I would think that they were thinking, okay, if there's evolution, then it all has to be good, and it all has to, I don't know, or they're thinking it has to be always something new, rather than integrating something from somewhere else into your genome. To me, it's what do you call it when you kind of cobble things together out of spare parts from different things to make another thing that's useful, at least for the time being. Like getting
junkyard engineering.
Yeah, just kind of.
Yeah. Yep. Okay. So then he's talking about how he mentions that they're more complicated. They have multiple proofreading steps, and their splicing of introns are tissue specific. They have a distinct purpose. They are not parasites, and their splicing of intron. Okay. So he's kind of suggesting that introns are functional everywhere, basically. And that was the first thing I asked him about. The claim that group 2 introns involved into the spliceosome is a big stretch. No, it's not. Not at all. He didn't read the papers. He's not thinking about the hypothesis itself, right?
And think about the big stretch of time in which it took place.
Yeah. The hypothesis, I had ChatGPT lay it out. We'll take a look at that later. But anyway, so Rebekah said, thanks for the great information. I'm pinning your comment. And then I said, introns in eukarya are also parasitic. I think you missed the point, because introns absorb all those mutations, right? And transposons. You'll find signs and lines just filthy inside of introns. I can show that to you. Oh, damn. Keep going the wrong way. Can I show that to you? Yeah, I can show that to you. Okay, where are we here? Okay. Okay, this is the gene right here. You see the stretch of the gene? It covers my whole screen, the FXN gene. If we go down here, what do we find? We find ALU, a line. ALU, another line. We find some signs down here too, I'm sure. And then we find these simple tandem repeats, which are also parasitic. And then we find the MER-E, I think it's a ERV, MER91C. All of these are parasites that are in the middle of that intron. You see where they are? They line up right here. So they're all in the middle of the intron. Oh my God, what happened here? Stop it. Okay. Do you follow that, Scott? You see all these viruses?
Yeah, my nose is starting to run and eyes are starting to water because of all the viruses.
Oh, okay. I thought so.
I think I caught something.
So anyway, so back to our conversation. Okay. So then intron, let's see, I said introns are also parasitic, and he's like, and then he says the evolutionary narrative is somehow connect self-splicing introns with the spliceosome. But spliceosomes are highly functional, and group 2 introns of bacteria demonstrably reduce fitness in most contexts. Okay. They reduce fitness, these parasites, right? And because they reduce fitness, if you have the parasite, there's going to be a small population somewhere of bacteria that have the antidote for that parasite. They're going to have the medicine, the antibiotic, right? Or the thing that's going to allow them to live with the parasite. And that's coevolution. So that's where things get interesting. And you take that coevolution for 3 billion years in bacteria, which can reproduce in 20 minutes. So they can have, what is that, 3 generations per hour. What the hell, 72 generations per day for 1 billion years. You can imagine how many generations. So anyway, let's see, only parasitic. Okay, let's see. Introns were considered parasitic in some contexts before. They now know they are highly functional. So he's claiming that introns, all of that, all of that junk that I showed you all.
I hear echoes of Donnie Deals in the word highly functional.
Yeah, exactly.
That's kind of a hand wavy thing because
it is.
The signal to noise ratio, is very, very low. The low signal, high noise in a lot of these examples, if I recall correctly. And then reducing fitness. Now, what is defined as fitness here? It could reduce fitness in some aspects, but then other benefits increase fitness. So I don't know.
Well, if they kill the bacteria before it divides, that's going to reduce fitness. But if the bacteria happens to have the antidote laying around in its junk DNA, it has a perfect antidote for it laying around somewhere in all that junk.
Yeah, but you never know what these ID people or creationist people mean by fitness. Is my comment.
Fitness is simply the amount of offspring that you have, basically per lifetime. Like your fitness, if you have 3 kids or 1 kid is 1.0, and mine would be 3.0 because I had 3 kids, and that's fitness. Anyway, okay.
But has that been proven? What has his statement been shown scientifically? Via evidence, via experimentation papers.
What's that?
These changes, you may have changed the, demonstrably reduced fitness in most contexts. Has that been tested? Has this statement been tested and found to be true? Or is this just a nonsense creationist thing?
That might be true. But I don't understand how they would find a bacteria that had that parasite in it that didn't have the spliceosome to save it from the parasite, because that bacteria would not have existed.
Caveat here, most cases, the majority demonstrably reduce fitness, but in some cases it doesn't, and that's where we find evolution, right?
Yeah.
Okay.
But in this, what he's saying here is that these parasites are junk DNA, and yet he denies junk DNA over and over and over again.
Gotcha.
Yeah, interesting. But he's saying if it reduces fitness, then it's junk. Deleterious junk, even worse than junk. Bad junk, the kind of junk you trip over when you wake up in the morning and you forgot to take all the beer cans out. That's
the stuff you throw it out onto the front porch because you keep tripping over it on the way to the beer cooler.
Yep. Unfortunate. Okay. They're only parasitic in the sense they were not functional. Unfortunately for evolution, junk DNA is basically a thing of the past. No, it's fucking not. It's because you don't understand it. Okay. Calling it junk on one side, calling it highly functional on the other side, both of those extremes don't make any sense in biology. Okay.
Yeah,
it's not.
What they did was take an unhelpful early adoption of a phrase, junk DNA, and decide, that's not a good term anymore. So we're going to call it non-coding.
Yeah.
What non-coding what? What do they call it? Non-coding
DNA.
Non-coding DNA. So
Yeah.
there we go. It's a better description, and it's not as misleading as early headlines and early adoptions of that idea.
Yep.
Made it.
So I call it junk drawer DNA, because you got all this stuff in your junk drawer. You got some Scotch tape in there. You got some razor blades. You got some, some washers, some old screws.
How many times have you put your hand in your junk drawer and gone, ouch, because there's something in there?
Several thousand.
Yeah.
There's a lot of blood in the bottom of my junk drawer.
Not a perfect fit.
Yep. So start making a case. This is my response. If you have this attitude about junk DNA, then you will have to tell me, for example, a 20,000 BP intron is good for in every case where we find introns. How do you justify functionality when we know that transposons, a form of T2 intron, extend introns, and other mutations are far more frequent and allowable in the introns? I showed you, basically one of the things that the introns are good for is absorbing random mutations, and I showed you that there were all kinds of viruses that jump into those introns. Those introns get bigger and bigger and bigger as the animal proceeds in its evolution.
So basically, if there's going to be a mutation, the statistical likelihood of a non-coding DNA segment gets the mutation is increased. It's like the DNA equivalent of a human shield.
Yeah. Yep.
Okay.
Yeah,
or pawns of mutation. They get mutated instead of the king or the queen or the knight.
I'm trying to think of an analogy. If if you've got if you got a garden and you got a lot of peas growing in that garden, right? And the deer come there and they just eat your peas. The deers and the rabbit and everything, they'll eat your peas, right? But if you had one pea plant in the middle of a whole bunch of weeds, the chances are that they would eat the weeds and miss a few pea plants, because it's just, statistics. It's simply statistics. Does that make sense? That's the only thing.
It lowers the statistical likelihood that damage is going to occur to something important.
Yep. God, I wish I could type. Okay. I prefer the term junk DNA. It all does something, even if that something is taking up time and space in a junk drawer. Okay. And then I mentioned the Dan thing, and then he came back. Sorry, I missed this comment, but I'm having difficulty understanding your post. I think you have a couple typos. Yeah. It seems that you're referring to 20,000 base pair introns. That was just an example, there are some introns that are 100,000 base pairs long. Some are 20,000, some are 5,000. But you didn't address the fact that spliceosomes are highly functional and algorithmic. Okay, there's the design terminology that he wants to use. I can't highlight things. Why can't I highlight things? Oh, there.
Image.
Yeah, I got my color set wrong on my Mac. Okay. The theory seems very incomplete, especially since it is thought by evolutionists that we evolved from bacteria, archaea, actually, with bacteria as mitochondria. But it seems your theory has huge gaps and a lack of mechanisms that can get from A to Z. Right. I'm going to give them the entire theory of spliceosome evolution, even though I just, started to study it myself in comments, right? So, I mean, huge gaps. They're always complaining about these huge, how in the hell are you going to, Rebekah, in the beginning of her alternative splicing thing, she had a paper that basically tells you what the hypothesis is. And it takes some doing to digest that paper. So find a source of this claim. You need elbow length gloves. Yeah. Yep. So right on, Austin. Okay. Do you have mechanisms that can fill in the gaps? Yeah, I fucking do. Otherwise, I have to call it speculation based on the idea of wide scale evolution is true. Oh, let's see here. what else we got? What else can we do? So his idea is that introns are all functional, that all of those random sequences are functional. And okay, so I looked into one of them, one particular, that FXN gene that I was telling you about, right here, repeat induced epigenetic changes in intron 1 of the frataxin gene and its consequences in Friedreich ataxia, which kills you. Okay. So it turns out that in normal humans, we have this repeat GAA, right? That repeats over and over. And it does so in normal humans between 8 and 33 times.
Is that Gattaca?
No. Well, yeah, that could be Gattic. There's no anyway. Okay, so but individuals in which they have 90 or more repeats in both alleles, they get this disease. So these repeats are things that, when you copy DNA and you have a repeating sequence, they're very likely to grow and shrink. Okay. So in people that have too many of those things, they get sick. They get this disease, right? That's in the middle of an intron. Now, that's hardly functional because it kills these people oftentimes when they're 16 years old before they reproduce. Okay. So this is just one example. And this is that gene. This is the one I showed as an example. And okay. So I went down and I downloaded that gene. Okay. And you see the capital letters here, right here. They're all caps. Scott, you with me?
Yes.
Okay.
Although I still, as soon as I start having to find the microphone, I can't find it anywhere.
Yeah. Okay, those are the exons, the capital letters. Okay, the lowercase letters here, these are introns. So I went down and I did a search and I found, here it is, this particular genome, which is the current T2T genome in UCSC, University of California, San Diego, something like that, Santa Clara. Okay, has a GAA repeated 9 times. They said 8 to 30 times. This one has 9 of them, 9 repeats. Okay, if we were to take that thing by some mutation and multiply the hell out of it, up to around 90 times, you would have a person that dies, basically. They die of intron. Okay. And what they found out...
Intron poisoning. Yeah,
what they found out about that is it's because of this thing over here. Okay. Now these TADS and LADS, okay, topologically associated domains, okay, and lamin associated domains are basically how the genome, or each of these colors here are chromosomes. Okay. So there's say chromosome 2, there's 2 copies of chromosome 2, and they occupy regions in the nucleus. Okay. But it's a little more complicated than that, because right here we have something that sticks to the lamin that's on the surface of the nucleus, and that's called a lamin-associated domain. And that means that DNA is probably not going to get transcribed, okay? But then we have all these little folds and mutations and things that stick together and stick out, and these are topologically associated domains. And this is where genome transcription happens. They have these little factories that produce genes, and depending on what kind of cell you're in and what time in the organism's lifespan you're in, you have that's heterochrony and heterotopy. You have these differ, right? And they differ because of these sequences, like right here, they pinch together. Okay. Let's see, right here, it pinches together because there's matching sequences that associate with proteins, and they basically create these folds. So in every cell, the reason cells are different is because different domains are topologically active. Okay. Now, what he is claiming is that introns do this, and lo and behold, they do it once in a while. We found a few cases, like I think a handful, like 20 of them, where the introns are the things that cause this pinch right here, right? That drag this DNA out. And then we have epigenetics too. So that's another thing that causes genes to either transcribe or not transcribe. He's claiming because we found this tiny little fraction of introns that actually affect this domain folding, that actually drag these things out here, that they're all functional. And creationists do that over and over again. If you find 20 ERVs that are functional, they're all functional. All, 10 million of them or 1 million of them or whatever. So is that clear? Or have I lost you totally?
Well, yeah, they're naming things as functional that probably have a much lower signal to noise ratio than real scientists.
Yep. Yeah. So what happens is if you have that mutation in that frataxin gene, basically one of these strands ends up sticking somehow, maybe sticking to the outside of the nucleus. You don't create that protein, your mitochondria don't work, and you die by the time you're, a teenager. So that is the case where a mutation, a copy mutation, kills you. Basically, it can happen in that one gene if you're unlucky enough to have that particular mutation. So that tells you that introns are highly unfunctional in that they can kill you. So in this one case, I found a case where it can kill you, basically. Okay. He is claiming that they're all entirely functional. They're just all blah, blah, blah. Over and over again, we run into this bullshit. Where did I? Okay.
In the area of functional, I just did a quick query with Copilot. So as always, go check for yourself independently. Young Earth creationists often define functional DNA as any sequence with observable biochemical activity or potential purpose aligning with a design-based view. Evolutionary geneticists, by contrast, define functional DNA based on evolutionary conservation and selected effect criteria, meaning the sequence has been preserved by natural selection because it contributes to fitness.
Right. So and if you look at
there's a big difference in the way it's viewed according to this. Now, I don't know what different creationists would actually say is their definition of functional, or maybe a lot of creationists would fail to define functional, hoping people will assign the desired definition to make it seem more plausible. I don't know.
If it was functional like creationists said it was, we couldn't possibly survive. Like Dan Gar said, Dan Gar, I think his name is. He said that basically, if that's true, then evolution is false and life couldn't happen. There's no way that life could happen, because in the course of your first few years of life, you'd reproduce your cells enough, have enough mutations that you would destroy yourself. Basically, there has to be junk. There has to be these non-conserved sequences of DNA. And if we look at introns, if I look at your introns compared to my introns, we're going to find millions and millions of differences. Between you and I, we have 3 million base pairs roughly on average that are different because those are not conserved. And yet you and I are both living, breathing creatures approaching our golden years, right? So obviously it isn't all functional. If it was all functional, you and I would have been dead by the time we were 2 years old. We'd have never reproduced. So I asked ChatGPT, so what has my creationist friend seen that leads him to believe that many introns contribute to TADS, topologically associated domains? some creationist article, or just that they use intron probes to find euchromatin? Now, that's the interesting thing. They use these intron probes. They take a piece of intron, which is really common, and they create what they call a probe, and they can do tests on it, and they can find out where the DNA is unfolded, where it's accessible, basically, with these probes. And he might have gotten, like, he looked at the 4D nucleome project, which is a thing that talks about how those domains, how those chromosomes are organized. And he saw something in there about introns being used as splices. Anyway, most likely a mix of half-digested buzzwords, 4D nucleome, TADS, introns, non-coding DNA is functional, filtered through creationist websites or videos that stitch them together without understanding what they mean. Let's separate the scientific reality from the creationist misread. What 4DN and related papers actually say. TADs are self-interacting chromatin regions, mega base, not the size of introns, but 1 million base pairs, right? Much bigger than introns. Scale blocks within which genes, enhancers, and insulators have higher contact frequency. And then they talk about, let's say they talk about the TADs a little bit. Let me see. Okay, 4DN researchers often use intronic RNA probes, RNA fish, to detect nascent transcription. And that's where he probably got it from, because those things, those RNA probes attach to that part of the DNA. Creationist blogs, Discovery Institute, Evolution News, and Views on Common Descent often quote legitimate findings about non-coding DNA being functional, add rhetorical glue. Since introns are non-coding, this proves they're functional, and 4DN shows they control 3D genome structure. Ignore that the same papers explicitly describe most TAD boundaries as protein-binding motifs and chromatin loops, not gene introns, right? So he saw one or two that do this, and he extended it. Oh, well, we still, we don't know anything yet. We're going to discover in the future that they're all functional.
Yeah,
and that's bullshit.
There's a weakness in the human mind. I think I've heard somebody call it unwarranted extrapolation. That
when
you take a few facts and then try to apply it to a wider area or a longer time span way out, you invariably get it wrong, because the extrapolation that you're doing is unwarranted given given the limits in our ability to measure and anticipate and things like that.
Yeah, case in point, our president sees a video on Fox News of Portland where we're up close and personal shooting basically some protesters that are getting rowdy in a half-block region, and he ignores the other 200 square miles of Portland that aren't on fire. You extrapolate. Right. So they cherry pick phrases like intronic enhancers contribute to 3D chromatin contacts, and then inflate that to introns build the genome's architecture, and they don't. The actual evidence linking introns and chromatin topology, a small fraction of introns harbor enhancers or boundary elements that may influence local chromatin contacts. Back to the frataxin gene, I showed you a paper that talks about a place where it influences those topological domains, but it ends up killing you, not helping you. And of course, there are cases where it ends up being part of your, part of what makes you you. So there are some functional ones, but probably a handful. There are, what the fuck are there? Something like 200 or 300,000 introns in our genome, and we found functionality within around 2,000 to 3,000 of them, basically so far. So that's 1%. Around 200,000. It ends up being 1%.
Okay.
So we found functionality in 1%, and of course you're going to, because that's how junk drawer DNA works. Okay. So the claim many introns contribute to TADS is partly true, but misleadingly framed. Some introns participate, but they are passengers inside larger architectural units, not universal frameworks. So, if someone predisposed to see design, 4D images look like blueprint structured, hierarchical, and regulated. When a paper says introns participate in nuclear organization, he hears every intron has a deliberate structural purpose. He's missing the mechanistic difference between emergent organization from physics and protein binding and intentional design. In short, your friend probably encountered creationist commentary on 4DN papers or saw figures using intronic probes for euchromatin and concluded these imply introns build TADS. Anyway, we're approaching an hour, Scott, and we don't want this to be long.
No, we don't. We both got things to do, and I'm sure everybody else does as well.
Yeah, there's a lot more.
Austin may be skipping some homework because he's watching.
Yeah. What is my okay, I always say things like the dipshit creationist fuck knuckles said. Okay, cool. Let's see what else we got here. Cebro is also a sitting for kibbles. What is that? So you probably got a bunch of this from Donnie Deal. Oh, sitting for kibbles.
I guess it's after money. Is that what he's saying?
Standing in poop he's talking about. I think he's talking about standing in poop.
Here we have another scientific observation.
Yeah, Jay, it is early as shit. This is kind of when my mind works. So, okay, so there's a whole lot more in here. And then he goes through this iPad example. I forget where the hell that is. But we'll talk about that maybe if we ever do this again. We're really good at promising to do streams and never doing them.
Indeed.
Yep. Okay, it's right here. If you went back into the time with an iPad and you turned on YouTube, how would people ascertain what they were seeing? And what if that person
on YouTube, they wouldn't see anything because there would be no YouTube.
He's saying that if you went into an ancient civilization and showed them an iPad, that they would say, oh, that has to be designed. A human had to do that. And no, that's not true. And that is a good topic for another lighter video.
Yeah.
That's less technical, because I want to address this. He keeps asking me, when are you going to address this? Well, I want to address it. So
he wants you to know when you're going to address this iPad thing.
Yeah.
Okay.
Yeah. This is his argument for, if you look at biology today, it's obviously intelligent design, and it's really an odd way to present it. But this
argument
is his
from vibes. He feels like it's designed, so it has to be.
Yeah, we'll get to it. Anyway, that's enough for today.
That's right, and I think we did a good thing getting logged on here and going over this stuff. It's very educational.
We should do this more often in the morning because I'm actually up in the morning. It's just that lately I've been working on Python coding and bioinformatics and a bunch of other stuff. Oh, by the way, let's see. Let me show you something here. Okay, before I go, I want to bait my friends. This is not available for non-atheists, okay? But I have a program. Where the hell is it here? Why won't this work? Oh, come on.
Because you're showing it off. You're
doing
a demo.
I want to show it to you. Let's see here. 100%.
Always problems just before a demo.
Yeah, you can make it a little bigger than that. I can go to 150%. Okay. So what this does is I have a thing that scrapes the comments, all of the comments for a particular video. You give it an URL, it'll scrape the comments, and it'll create a really nice spreadsheet formatted list of all the comments. Also
write some code to do a diff, like keep samples of the comment scrapings, and then do a diff so that you only see the new comments.
Yeah, that's what I need because it takes a while to run this, and there's a danger of being called a bot because but this column here
database and keep because you're going to have to run it in. Do you specify just your channel or just a particular video?
Particular video.
Yeah,
any video.
So you're going to have to manually run the thing anyway. So
right. This column here tells you which comment it is. Here's comment 1, comment 2, nobody responded. 3, you have 2 replies. 4, you have a whole bunch of 3 replies. And, so it's really nicely laid out. It tells you whether, if it's I think this is a primary. This is a reply. It says true, it's a reply. False, it's not. True, true, true. You see how it is? Anyway, I can run that for. I should charge people $5 every time I run it though, because I got to get paid for this shit. That was quite a bit of work.
So you just have people send in their information, and then you just bill them every time you run it.
I should. Anyway,
I'm sure you'll get a lot of takers for that.
But if you don't want to pay me, reach out to me, and if you're interested in capturing comments, I can help you out with that.
I used to get so excited when I would get a notification that somebody replied to a comment I made. Now I never get those. Yeah,
you don't get notifications anymore. That's why I don't know how they think that's going to work, because people are going to miss all kinds of comments. It's just
you don't get to argue back and forth unless for some reason, and this may be like trying to reduce traffic or something for practical reasons, but now you have to remember all the threads that you're commenting in and go back and check for people replying to you and then replying to your replies.
Yeah. And I'm going to lose interest after about 4 days, and I'm never going to look at the video again. So anyway, so I linked Rebekah Rong's video. That's what I'm going to call her from now on, Rebekah Rong, RR, in the description. And I linked that one paper that I looked at. And I don't know. Anybody has questions, reach out to me. I hope I explained introns to you, Scott.
Yes, you have. You have in the past, and you did today. Eventually, I'll be able to get it.
I'll do it again next week. Okay, good enough. Okay, so I think we're out of here. Thank you guys for listening. All 2 of you.
Everybody have a delightful morning, evening, or night, whichever your time zone is, and
yes.
keep it going into the weekend too. Just have a great week.
Okay. Goodbye, cruel world.